Thank you very much, Sven, for the introduction, and thanks to Patri, James, Sven, and Gustav for inviting me. I'm very excited to be the last participant, and I'm also very excited to be part of this all-female session in the Hydrogenase seminar. And I'm going to continue talking about structures, and we're moving also to an oxygen-sensitive enzyme, but I will talk about the iron-iron hydrogenase from disulfuribuldisulfuricans. So first of all, let me take a step back and tell you a little bit about our motivation to study enzymes that are capable of producing hydrogen. So hydrogen is considered an ideal energy carrier because it has a very high energy density. And I recently found this number that 1 kilogram of hydrogen gas is comparable to 2.575 kilograms of gasoline. It also has a clean combustion, producing only water as the product. Currently, hydrogen gas is produced from fossil fuels or by water electrolysis. And the energy for water electrolysis is also mostly derived from fossil fuels. So we're looking for a more sustainable way of generating hydrogen if this is really a potential option for energy carrier in the future. Of course, nature has already solved this problem, and there are naturally occurring enzymes that are capable of producing hydrogen. These enzymes operate at high rates, high efficiency, and under physiological conditions. So these enzymes, the hydrogenases, catalyze the reversible 2-electron oxidation of molecular hydrogen. They are involved in processes such as nitrogen fixation, microbial phosphorylation, and fermentation to methane. They occur in anaerobic and some aerobic microorganisms, and their primary function is to provide energy for the organism. So hydrogenases catalyze the reversible reaction that consists of reducing equivalents by producing hydrogen or providing electrons by splitting hydrogen. These enzymes are of biotechnological interest. Potential applications that hydrogenases could be used in the future are microbial degradation of organic material, the production of methane, and of course, the generation of hydrogen as energy source. So the concept that nature uses for hydrogen splitting is that the reaction occurs in a heterolytic way. The acidity of dihydrogen is extremely low with a pKa of 35. So the binding to the metal side dramatically increases the acidity of hydrogen and enables the splitting reaction. So because of the binding to a metal, a 2-electron transfer is feasible at physiological conditions, and the inorganic components of the hydrogenases serve as electron reservoirs and the catalytic centers. So the toolbox that nature has are 3 distinct classes of hydrogenases, and the classes are divided according to the metal cofactor. So we have nickel-iron hydrogenases. We just heard about nickel-iron hydrogenases. They consist of, the active site consists of a 4-iron 4-sulfur cluster and a nickel-iron cluster. So this bimetallic cluster is similar to the iron-iron hydrogenase, which has an iron-iron cluster. And there is a 3rd class of hydrogenases, the so-called iron-only hydrogenase, with only 1 iron in the active site. So if we're looking at the bimetallic hydrogenases, we see that the hydrogen binding site is between the metal centers in nickel-iron hydrogenases. They are less oxygen sensitive and they're biased towards splitting hydrogen. Iron-iron hydrogenases have a different binding mode for the substrate. So hydrogen binds to the distal iron side, and they are highly oxygen sensitive and biased towards evolving hydrogen. So now I would like to introduce the hydrogenase that we're interested in, that we are studying at the moment. Disulfuribuldisulfuricans hydrogenase comes from an organism that is gram-negative and a sulfate-reducing bacteria. The iron-iron hydrogenase is highly active, especially in terms of hydrogen production. And the crystal structure has been reported approximately 20 years ago. And I would like to quote Oliver Einsle, every protein wants to crystallize. And I think this is particularly true for this hydrogenase. And the structures are beautiful at very high resolution. So this structure was solved to 1.6 angstroms, if I recall correctly. And the overall architecture, you can see that we have a large subunit in gray that binds all the metal cofactors. And we have a small subunit in teal that is wrapped around the large subunit. The small subunit does not contain any cofactors, but it's kind of wrapped around the protein. If we look at the active site, it's buried deeply into the heart of the enzyme, and we have a so-called H cluster that consists of a Cuban type 4-iron-4 sulfur cluster that is coordinated by 4 cysteines, and one of the cysteine is bridged to the 2-iron subcluster that also contains carbon monoxide and cyanide ligands and an ADT bridge. In addition to the H cluster in the active site, we have 2 4-iron-4 sulfur clusters that are called the F clusters. They're responsible for efficient electron transfer to the active site. So if we look at the mechanism of hydrogen splitting, to activate the small molecule hydrogen, a low valent metal is required that functions as a Lewis acid, and a pendant base, a Brønsted base, is required that is provided by the nitrogen at the top of the ADT ligand. So we have the distal iron and the ADT nitrogen that activate hydrogen. I thought about what to put on the slide for the proposed reaction mechanism. The only conclusion that I could think of that we all agree on is that the reaction mechanism remains a topic of controversial discussion. There is an active oxidized state, H-ox, and an active reduced state, H-red. Both of these states have been characterized structurally, although it is a little bit difficult to say sometimes in the crystal structure if you have only an oxidized state or if you have a mixture of oxidized and reduced. So with our methods, we intend to add a little bit more to this, more knowledge to this field and to the reaction mechanism of iron-iron hydrogenases. And these are 3 of the questions that we are interested in answering with the methods that are available in our lab and together with our collaboration partners. As Sven mentioned, a very good choice. So we would like to look at which intermediates can we identify in the active cycle. So far, there are several remaining intermediates that have not been characterized structurally. How does the inactivation and reactivation work? So this is this pink arrow that starts with H-ox and goes out. I will talk about that a little bit in more detail. And how does CO inhibition work? So the methods that we use in my lab are X-ray crystallography. And we also have a similar setup that Bonnie showed previously with an ECTA system that we can isolate proteins in an anaerobic environment. We can also crystallize, and we have a microscope in our glove box, so we can prepare the crystals and we can freeze the crystals under anaerobic environment. In addition to X-ray crystallography, together with our collaboration partners, we also perform spectroscopy in solution. And we prepare crystals that are then measured for single crystal spectroscopy for our collaboration partners, and we'll show some of that data later. And we also combine the structural knowledge, the spectroscopic information with computational methods to provide a complete picture of what is going on in this enzyme. So the so-called inactivation sounds a little bit weird that people are just studying an inactivation. We're not particularly interested in how and why the enzyme is inactive. But this enzyme for desulfurylglycans can be isolated in an oxygen-stable inactive state. So the oxygen-stable properties are, this is what is really interesting for us, because as I mentioned, these enzymes are highly sensitive to oxygen, and it makes it less attractive for applications if you have to prepare devices under anaerobic conditions. So there's this very nice experiment performed by my lovely collaborators, Patria and James, and they used hexamine ruthenium chloride as an oxidant and treated the enzyme in the presence of oxidant and a sulfur source. So what they were able to show and detect using FTIR spectroscopy is that starting from the HOX, so the active oxidized state that you can also see here, if they treat the enzyme with only oxidant, we don't observe a lot of changes. If they add a sulfur source by itself, sodium sulfide, we also don't see any changes. The enzyme is still in the HOX state. But if we combine oxidant and a sulfur source, we see slight changes to these yellow peaks that are named H-trans, and further addition of oxidant then forms the H-inactive state, the oxygen stable and inactive state that we are interested in. And this is not a dead state, so the enzyme is in this state can be reactivated under reducing conditions to yield then the active oxidized state again. So the first goal that we had in our collaboration is to study how this inactivation works. And this all started as usual with a beer in front of a poster at a conference and me asking why they did not crystallize it yet. So the next step was then that they sent enzymes and we crystallized it. And just a few background information, the enzyme was produced recombinantly and maturated artificially and then transformed into the H-inactive state. We crystallized the enzyme within 3 days under aerobic condition, because this is oxygen stable for approximately 3 days. This is what we know. And the crystals or the protein crystallized in the orthorhombic space group P212121, and we were able to solve the structure at a resolution of 1.65 angstrom. So this is one of the prettiest crystal that we obtain. This is the setup here for X-ray diffraction experiments. This is how a typical diffraction pattern looks like. Nowadays, the detectors are so powerful that it's really hard to see the diffraction pattern at all, but luckily the software recognized the spots and is able to calculate the structure. So when we solved the structure, the first thing we did is to compare our overall structure to the previously solved structure. And we see that the overall architecture is very, very similar. We also see the cofactors, so the H cluster and the active site and the 2 F clusters, and we see that they are fully intact. One thing that I have to mention is that in the active site, we see, so the 2-iron subsite that was maturated artificially is only 60% occupied in the structure. So then we were still interested in why the enzyme is not sensitive to molecular oxygen, or what is the mechanism of protecting the active site against molecular oxygen. And we did X-ray diffraction experiments at 12 kilo-electron-volt. And at this energy, the data can be used to calculate the native electron density map. So this is all atoms minus the hydrogens. And this map allowed us to identify an additional ligand at the open coordination site. But we don't know anything about the nature of this additional ligand. So then we collected X-ray diffraction experiment at 6 and 7 keV, and this enables us to determine the anomalous density map. And at this energy, only sulfur or chloride ions are possible elements. So we can calculate an anomalous map that shows the substructure of either sulfur or chloride at the same time. So we cannot distinguish between these two elements. However, we combine this with other techniques, and especially X-ray absorption spectroscopy was also consistent with the presence of an additional sulfur ligand. So this is the anomalous density map, and I only showed this for this extra ligand. Of course, we also see density for the cysteine and the other sulfur, bridging sulfurs, so all the sulfurs in the structure, thymines, cysteines, the 4-iron 4 sulfur cluster. So we see electron density for all these atoms. What is also really nice is that at 7 keV, right below the iron edge, so we don't see the electron density of the irons that could sometimes maybe mask this electron density because they're very intense. So 7 keV actually turned out to be the perfect energy for determining the sulfur map. Yeah, so this is the picture that we then modeled. As Bonnie mentioned, the electron density is the blobs that we get as crystallographers, although this is actually a very nicely electron density, so it was not that hard to model, to build the model. And in the end, the crystallographer or the structural biologist determines or prepares a model, and this is what you then can download in the PDB. So you always have to keep in mind that the models that you get, the positions of the atoms, have always a bias of the researcher that builds the model. So we determined using X-ray crystallography that we have a sulfur species or a thiol binding to the substrate binding site. We also have additional information, spectroscopic data on the HNACT state. We have, as mentioned previously, X-ray absorption spectroscopy, FTIR spectroscopy, NRBS. We complemented the spectroscopic studies in solution with quantum mechanical and molecular mechanical calculations. And we also performed spectroscopy on the crystals. So usually this is always a problem that we have data on protein in solution, and then we have a crystal structure, and we're not entirely sure what state our crystal structure. We know the state of the protein when we set up the crystals, but 3 days later, we don't have a good method of determining that. And with the development of single crystal spectroscopy, we actually have the tools on hand to determine the state of the enzymes in the crystal. And this is work done with Christian Lorent and Ingo Sepka at TU Berlin, Technische Universität. And he was able to characterize the crystals that we sent as the HNACT state. So this was our first goal, to characterize the mechanism of inactivation. And now we're also interested if we can reactivate the state, and in particular, if we can reactivate the state in the crystal structure. So crystals consist of 40 to 60% water, so they are quite flexible. So I already learned in my PhD that chemical reactions can occur inside a crystal. So what we did, we crystallized the enzyme in the HNACT state under aerobic condition, and then we tried to activate it in the presence of sodium dithionide, a reductant, and hydrogen gas. So hydrogen gas approximately 2%, because this is in our glove box. And what we could actually see is, so this is the structure that we start off. This is obtained under aerobic condition, and then if we introduce it into the glove box, so we set up the trays right in the glove box, seal them, wait 3 days, and then harvest them in the glove box, we see intermediate states where the ligands partially disappears, and we have some structures where we actually see the electron density completely gone. So in most of the structures that we collect, we see an intermediate where we see low occupancy. So we can determine how much ligand is still left in compared to the other atoms in the 2-iron subcluster. But we can also see the ligand fully disappearing in the crystal structure. And this was potentially exciting because we think that this way we can crystallize H-intact under aerobic condition, and under anaerobic condition then address different states in the catalytic cycle. Because reaction doesn't have to stop with Hydrox, but we can also try to push the reaction forward and characterize different structures. And this is very nice because then we know that these are on the catalytic pathway and not just some artificial states that we generated. So as I already mentioned, we are also trying to characterize our crystals using single crystal spectroscopy, and we collected for several crystals that we set up under these conditions IR data. This was also done by Christian and Ingo in Berlin, and we tried to freeze crystals from the same batch for IR spectroscopy, and or we not only tried, we were able to freeze crystals from the same batch. And these crystals were obtained under 2% hydrogen and treated with sodium dicyanide in the dark, because we see some damage if we expose the crystals for an extended period of time to light. And Christian's data actually showed that we are not in the H-intact state anymore, but we activate the enzyme to a mixture of different states. Some of them are H-trans. So H-trans still has a ligand. So this is not quite in agreement with our ligand disappearing. So potentially, we also have some help from synchrotron radiation to get rid of the ligand. We don't know. We also see a small portion of CO inhibited enzyme. This also means that we expect a diatomic ligand, and we see evidence for HOX and HRED plus in the IR spectra of the crystals. So we're still working on optimizing these procedures and trying to make the measurements of the IR spectroscopy crystals as similar as the crystal structure, but I think that we have sufficient structures that we can now say we can actually reduce the H-intact state and reactivate it to HOX. And the next step that we looked at is the CO inhibition. So spectroscopic studies have suggested that the hydrogenase can form CO inhibited states. And this was previously also shown for CP1. And we tried to crystallize the HOX state this time. It also crystallized very beautifully in the glove box. And Christian borrowed us this nice chamber where we can put our crystal trace in and then expose it to CO gas. So this is the resulting crystal structure that we obtain. And with in the electron density map, we see evidence for a diatomic ligand. And we propose that this is the exogenously added CO that binds into the substrate binding site, so the same site where the thiol binds, or the hydrogen substrate. So after we learned a lot about the inactivation and reactivation and the CO inhibition, we also want to learn more about the reaction mechanism of this fascinating hydrogenase. And therefore, we tried to understand the role of some amino acids in catalysis, and two variants were created, the C178A and C178S, and we solved the crystal structures for these two variants. And interestingly, they are also not sensitive to oxygen. And we see in the C170A variant that we have a diatomic ligand. And we have also more data that supports an additional cyanide ligand in this active site. And we also have a structure of the serine mutant, but the spectroscopic data are not very clear, and we modeled a water because we're not sure what this monoatomic ligand is. But yeah, we're still working on this. And for the C170A mutant, we proposed the formation of H-intact-like and H-trans-like states. So with this, I hope that I could show you that DDH likes to crystallize and that we can address various states with a combination of crystallography and single crystal spectroscopy. So we're particularly interested in this hydrogenase because it's highly active hydrogen-producing enzyme, and we're combining X-ray crystallography with single crystal spectroscopy to determine the state of the enzyme in the crystal so that we really know what we're looking at. So far, we've been able to show that DDH can be inactivated to form the H-inert state, which is stable towards oxygen. The state can be reactivated in the presence of sodium cyanide and hydrogen. We also could show that or determine the structure of H-ox that have been treated with carbon monoxide, and we show that the CO ligand binds to the substrate binding site, and we see that a variant, the C178, binds an additional cyanide ligand at the open coordination site. So taken together, we have studied the interaction of the hydrogenase with a lot of small ligands, and we hope that we can use our expertise in the future to understand the active cycle of iron-iron hydrogenases by correlating three-dimensional and electronic structures. Last but not least, I would like to thank my students. So I still have some students in Düsseldorf. Former students Lisa and Hannah contributed to this project, and Melissa and Nina, but most of the work was done by Konstantin. I'm very happy that he moved with me to Erlangen, and this is the first group picture at Bergkirchweih. I can highly recommend to visit Erlangen around this time of the year, and my new students, and I would like to thank my collaborators at the MPI for chemical energy conversion, Serena, James, and Ragnar. Patri was at the MPI when we collaborated and now moved to Oxford a while ago. Ingo at Christian and Charlotte from the TU Berlin, and DFG, Fonds der Chemischen Industrie, and Jülich and the Ministry for Innovation and Science for funding. Thanks very much for listening, and I'm happy to answer questions.